Hey, Mr. P here. We are out of DNA structure and packaging, and now we're into DNA replication. So we need to look at how DNA replicates itself, meaning how are the enzymes that are essential in DNA replication utilized, what is the order that those enzymes are utilized in or used in, and how do those enzymes work together in order to replicate a pretty complex molecule in DNA. So we're going to look not only at eukaryotic DNA replication, but we're also going to look at prokaryotic DNA replication. For the purposes of IB, at least at the HL level, we are going to focus on prokaryotic DNA replication, but both mechanisms are pretty similar, and so DNA replication is kind of generalized DNA replication. So the first thing I want to do is talk about the modes of DNA replication. Now we know from our studies of biology that there were three main ideas on how DNA was believed to have replicated or is believed to replicate, and so the first is this conservative model. Alright, the second being a semi-conservative model, and the third being a dispersive model. Now we know just from our first videos or our prior videos in the structure of DNA that the semi-conservative model is the correct model in that our original strand, which can be seen here in dark blue, is going to be split apart. Basically, if we look at this, it's going to produce this replication fork, and I know we haven't talked about how the replication fork is produced yet, but we will. And by splitting this molecule in half, you're using the two dark blue template strands to produce light blue complementary strands. Okay, and so you can see that in both of these new strands, there is a dark blue and a light blue. The dark blue being an old strand, the light blue being a new strand. And so that's semi-conserved. The old strand, which we can see here, the original strand, is conserved semi or partially conserved in both of the new strands, and then those go through replication again. Again, this old blue strand is here. The light blue strand would have been here or here, but either way, it would have been split up, and so new strands would have been built and would have been complementary to these two template strands. This is how DNA replication works. That is a semi-conservative model. Now, the two models that are incorrect are this conservative model, and that's obviously, or it's incorrect for obvious reasons. This being the old strand is in this model going to basically sit there and be copied from a distance, meaning the entire old strand is going to go to the left in this diagram, and this entire new strand is going to be produced basically by looking at this old strand. So it's almost like a sketch that an artist does by looking at an object, and we know that there can be or are going to be errors associated with an artist sketching an object. Even if you're a really good artist and even if you can draw really well, you're probably not going to have the exact proportions correct when you do a drawing like that, and so it's very easy to understand that if we have an entire old strand that is kept together and intact, and we build this entirely new strand from it, there's probably most likely going to be errors, which is going to lead to potential problems when it comes to potentially cancer or other genetic disorders like that. So conservative model is incorrect because, again, there is no using of template strands like there was in this semi-conservative model. Now, dispersive model is absolutely false, and the reason it's false is because in the dispersive model, we are essentially taking our template strand and we are splitting it up. We're dicing it up. We're sending some of the old DNA in different sections to this left side, and we're sending some of the DNA in sections to this right side, and we're filling in the gaps with new DNA. Okay, so we would have the new DNA here in the light blue, and we would fill in those gaps. Like I said, the old parts over here are the same new parts over here, and so we never want to cut our DNA into pieces like this, because you're just asking for mutations and other errors, which obviously is going to lead to cancer or lead to other disorders. So what we can kind of collectively agree on is that this semi-conservative model is correct, and these are the reasons why. How is prokaryotic DNA replicated? We know from prior sections of the IB syllabus that the prokaryotic DNA is naked, and what we mean by that is it is not associated with proteins. Those proteins obviously referring to histones. And so prokaryotic DNA does not contain histones. It is not supercoiled. And it is kept in a circular ring called a plasmid. And it is not linear like eukaryotic chromosomes are. So if we look at this ring of DNA called a plasmid, it's a circular ring of DNA, not associated with proteins, we need to look at the origin. We have an origin of replication, which is in this diagram right here at the top. There is a box around this origin, and so from that origin, in any bacterial plasmid or bacterial DNA, replication is going to happen in both directions coming out of that origin. The origin of replication is where replication is going to start, and that's the reason it's called origin. Okay, so that's the starting point for DNA replication. The DNA helicase, and we're going to get to these enzymes, but basically helicase is going to travel in both directions equally, and the reason we can increase, or the reason we can replicate the DNA in both directions is obviously because of speed. We want to be quick, we want to be efficient, we don't want to waste time, and so if we can work both directions equally, we're going to essentially start here and end here. And so you can see as this time lapse proceeds, we're going to start at our origin, which is here and here, and we are using the template strand, this being the template in both cases, to produce the new complementary strand. And so this portion of this strand here is going to be complementary, exactly complementary, A pairs with T, C pairs with G, of the template strand. Okay, it's going to proceed, it's going to keep going in this direction, and eventually it's going to terminate at a termination site. And after it originates here and terminates here, we have successfully completed DNA replication, and we have produced via semi-conservative replication 2 new strands of DNA, or in this case 2 new plasmids, circular rings of DNA. So now let's look at how eukaryotic DNA replication happens, or how it contrasts with the prokaryotic DNA replication. We know from that previous slide that DNA replication in prokaryotes is going to be in the form of replicating that plasmid. We know that prokaryotic plasmids or prokaryotic DNA are kept in rings, and eukaryotic replication is going to be kept in linear chromosomes. And so this would be how chromosomes or linear sections of DNA are going to replicate. Instead of having a ring that has one origin and one termination, we're going to have a linear strand of DNA. And because we have a linear strand of DNA, which in a lot of times is way bigger than the bacterial plasmid, we have to replicate it at multiple sites in order to increase efficiency and speed up this process. And so if this is our entire DNA, linear DNA, this entire chromosome, we're going to not only originate it in the middle, but we're also going to originate it here and here, and replication is going to happen in both directions from each of the origins. So notice we have an origin of replication number 1, origin of replication number 2, origin of replication number 3. And as we progress this, the origins are going to, or DNA replication is going to be producing the new template or the new complementary strands, which are the light blue in this case. Okay, these are the new complementary strands using the old strand, which can be seen in the dark blue. So this would be the old, these would be the new. And again, this is just showing you semi-conservative replication. but in a linear strand of DNA, it's showing you that there are multiple origins on a eukaryotic chromosome. Okay, it's really important. Multiple origins on a eukaryotic chromosome. These molecules, these linear chromosomes, are just way larger than bacteria or prokaryotic plasmids. Okay. How does that look on a chromosome? If we were to zoom out from that one section of DNA we just looked at and look at the entire chromosome, we know that in the middle of a chromosome somewhere is a centromere. We know that centromere to be the molecule that essentially holds two sister chromatids together, forming the entire chromosome. And in this case, there's going to be an origin of replication here, 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 and here. And so if we can open up these DNA molecules using enzymes, and we can start replicating here, 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 by the time we progress this, these are going to work out of the origin in every case. And because we are opening up multiple sections, we can completely replicate this entire strand of DNA in 5 times less amount of time, okay, or 5 times faster, because we are opening up 5 times more origins of replications. If we just opened up one site here, this replication would have to travel the entire thing. Obviously, this distance that I just drew is much longer and would take a lot longer than all these small arrows that are happening at the same time. Okay, so this is how it happens. Again, this is eukaryotic DNA replication. Very different, but very similar, right? We still have an origin of replication. We're still going to have replication kind of going out of the origin on both sides, okay, right to left or up and down. And so now let's look at the specific enzymes associated with replication. I'm going to kind of step these in order, and so I've got these steps out here. So number 1 is going to be the first, and then obviously on this slide, number 4 is going to be the last. But the first thing that happens, and I know there's a lot going on this slide, but just kind of take it step by step by step. It's very simple. Step number 1, at this origin, and so this is the origin of replication, we are going to open it up using proteins. Okay, every step along this DNA replication metabolic pathway, we're using enzymes, we're using proteins, and so proteins and enzymes are essential to do any kind of metabolic activity, and in this case they are essential to replicate the DNA. And so we're going to start with an initiator protein, and that protein is essentially going to allow for the enzymes helicase and gyrase, which also is known as topoisomerase, to bind to the right origin or the correct origin of replication. Okay, so this is going to essentially prime the system, so to speak. It's going to give these initial enzymes a place to bind. And so that kind of prepares this DNA strand, kind of loosening it up. It prepares it to slightly unwind. It gives a little bit of separation, which is critical in order for these two enzymes to successfully bind. If these enzymes don't successfully bind from the get-go, this entire DNA replication process is going to not happen correctly. So this step is essential. Prime the system, prime the parent DNA, loosen these up, break a few hydrogen bonds, give a little bit of energy. Okay, we have to use a little bit of energy, but again, the amount of energy we use is very minimal in terms of, or a good investment in terms of why we need to replicate the DNA in the first place. Once we have primed the system, we are going to allow, step number 2, we're going to allow DNA gyrase and DNA helicase to bind. DNA gyrase is going to be number 1. DNA helicase is going to be number 2. DNA gyrase is going to lead the entire metabolic or the entire enzymatic series of reactions. It's going to travel down our DNA strand. It's going to be unwinding the DNA double helix. Remember, before this, or on the upstream side of this enzyme, this DNA would actually be twisted in the form of this DNA double helix. So instead of looking at the DNA as this long, straight, linear molecule, let's think about it as a actual twisted ladder or a double helix like we know DNA to be. Okay? What is the job of the DNA gyrase? Again, it is to unwind this so that it's a nice even train track, so to speak, for DNA helicase to travel along right behind. And the job of DNA helicase is to break the hydrogen bonds. Okay, and so this DNA gyrase is going to straighten out the DNA molecule while DNA helicase follows along and breaks the hydrogen bonds. As soon as the DNA helicase, and this is all kind of one specific step, as soon as the DNA helicase breaks these hydrogen bonds, remember it's consistently moving, and so these single-stranded binding proteins, SSBP, single-strand binding proteins, is going to, or are going to jump onto the strand, and they're going to hold the DNA strand open. And how they do that is they disallow or keep the H bonds or the hydrogen bonds from reforming. Okay. If helicase was allowed to break these hydrogen bonds, which they are, or which it is, and you didn't hold this strand open by the single strand binding proteins, then right behind this helicase, as it consistently moves down this molecule, the hydrogen bonds would just reform, and you would essentially be zipping or unzipping and zipping the DNA molecule at the same time. Okay, step number 3. Once the single strand binding proteins jump on and hold this replication fork, which is what this is, replication fork, once the replication fork is held open by these single strand binding proteins, RNA primase is going to lay down RNA primers, which is going to allow DNA polymerase 3 to bond. So notice we have a series of reactions. One has to happen before another, which has to happen before another, which has to happen before another. First, we had the gyrase unwind the DNA. Then we have DNA helicase unzipping the DNA. Then we have single-strand binding proteins holding the replication fork open. Then we have RNA primase laying down these primers. Now you notice in red that these are the RNA primers, and so we're just going to say there's a few little RNA primers. Typically, there's anywhere from like 5 to 9 RNA primers. Those are actual RNA nucleotides. They have to be changed, though, to DNA later, and we'll talk about that. So this is the actual RNA primers, which are placed by RNA primase, which is basically producing a start site for step 4, which is DNA polymerase 3, which you'll notice these were the primers, okay? These were the RNA primers, and DNA polymerase 3 is now putting the DNA nucleotides in a 5' to 3' direction. But the job of DNA polymerase is to build the DNA molecule by placing the DNA nucleotides. Okay, so it's placing those DNA nucleotides 5' to 3'. Notice that this is the 5' end of the molecule. It's placing the new nucleotides one by one by one, and it'll continue to do that all the way along as this replication fork moves to the right. And if you're constantly placing these DNA nucleotides to the 3' end, you are building the new strand from 5' to 3'. And so, 4 steps so far. Very important to understand all 4 of those steps. Then we go to step 5. Step 5, RNA polymerase 3, or DNA polymerase 3, is continuing placing the DNA nucleotides, okay, in a 5' to 3' direction. Notice that these were the primers. We are continually placing these DNA nucleotides. Notice that on this one it's longer. This is continuously happening. This, by the way, is called the leading strand because it is built continuously. Okay, this one's built continuously. As soon as you place those RNA primers, you're going to start building the DNA molecule, and notice that this one is never going to stop. As this replication fork continually moves this way, this DNA polymerase 3 is going to just continue moving with it. It's never going to stop. It's never going to be interrupted. It's made continuously. That is called the leading strand. This one, however, because of the way of this, or because of how DNA is anti-parallel, because DNA is anti-parallel, we know that the strands run opposite of each other. And so if this strand is being made continuously 5' to 3' from left to right, we know that this new strand has to be made this way from 5' to 3'. DNA is always made 5' to 3'. Always. Okay? Always. So we always have to make the new strands of DNA 5' to 3'. That didn't come out right. 3'. This would be 5' to 3' in this direction. Now, what has to happen on this particular strand? By the way, this is called the lagging strand. We have the leading strand, we have the lagging strand, and the reason it's called the lagging strand is because this strand is made discontinuously. Jeez. I need to quit touching. Okay, lagging strand, because it is made discontinuously, or it's made in fragments, and those fragments are called Okazaki fragments. We're going to get to that, Okazaki fragment number 1. But how is the Okazaki fragment made? We still have to place the primers, and so what happens is the RNA primase comes in, lays the RNA primers here at number 1, and then it skips ahead a few thousand base pairs and it lays more RNA primers. Okay, and then it's going to skip ahead and it's going to make more RNA primers, and then it's going to skip ahead and it's going to place more primers. Notice though that when it skips ahead like that, it is moving this way. Okay, it's moving this way overall, but these DNA polymerase 3, just like it did here, DNA polymerase 3, notice the same shape, same color, DNA polymerase 3 still starts at the RNA primers just like it did down here on the leading strand, but because we have to build 5' to 3' going back to the left in this case, it's going to start at the primers and it's going to build the DNA molecule until it ends at the previous primer. Let's say we had primers here, and then it's going to fall off. It built this fragment, okay, that's a DNA fragment, and then it's going to jump ahead to these primers, and then it's going to build again in a 5' to 3' direction to these primers, and then it's going to jump ahead to these primers, and it's going to place the DNA nucleotides, and so you can notice that it's building these fragments in a 5' to 3', but it's discontinuously. It's making fragment and fragment and fragment, okay? That is DNA polymerase 3. Then DNA polymerase 1 is in charge of coming in. Notice that it's still the same color, but it's a slightly different shape, right? DNA polymerase, it's still the same type of enzyme. It still has the same type of job, but the DNA polymerase 1 is in charge of removing the primers and replacing the primers with DNA nucleotides. So notice that in this case, it's taking the red primers out and it's going to place DNA nucleotides in its place. And so it's going to go here, and then it's going to go here, place DNA nucleotides, and then it's going to jump ahead, place DNA nucleotides, and so that's how we switch those RNA primers, which were needed to give DNA polymerase 3 a starting spot, but we don't want to have DNA and RNA together because we are replicating DNA. We want the entire molecule to be DNA. We have to switch those RNA primers to DNA nucleotides, which we do with DNA polymerase 1. But the last step, okay, and that was step 6, the last step is step 7, and that's when we employ DNA ligase, which is our welder, okay, and I say welder, it doesn't actually go in there with a spot welder or a TIG welder or whatever, but it is tasked with actually linking the fragments. And how does it do that? It produces those phosphodiester bonds which links the backbone into a consistent strand. Okay, we want to make those discontinuous fragments into a nice uniform nice uniform strand. Okay, and so by the time primase and DNA polymerase 3 and DNA polymerase 1 and helicase, by the time they go all the way through, we're going to not only have a continuous strand down here on the leading strand, we will have a continuous strand running the opposite way on the lagging strand. Okay, again, these are called Okazaki fragments, named after Mr. Okazaki or Dr. Okazaki, who was instrumental in discovering those. And so these are the steps of DNA replication. The only thing we need to do now is just a kind of a quick sum up. And so this is a diagram of a replication fork. Okay, and so replication fork, which is only half of the bubble. The other fork would be over here. Remember, it runs out of the origin of replication, which we will say was maybe here. And so replication would have been happening that way and this way, but we're only looking at this half of the DNA replication bubble. And so the first thing that has to happen is, you guessed it, the DNA gyrase, which is green in this case, is moving down this strand of DNA, and it is relieving the strain, and it is relieving the supercoiling. It is uncoiling the DNA as it goes. The next step is the helicase. That helicase, which is here in pink, is traveling right behind the gyrase, and its job is to break the hydrogen bonds, which is critical to actually open up the DNA double helix. Okay? After the DNA double helix is unzipped, okay, or after the replication fork is opened up, the single strand binding proteins are going to keep the hydrogen bondings from forming. And that's going to prevent the hydrogen bonds from reforming, and it's going to allow the replication fork to stay open. The next step is RNA primase. Now you can see the RNA primase down here, but we know from what we just talked about on this leading strand, because remember this is the continuous one, that the primase had to be up here as well. So primase is laying the RNA primers here and laying the primers here. It's needed in both strands. The DNA polymerase 3 needs to have the RNA primers placed in order to give DNA polymerase 3 a starting spot. Once those primers are placed, DNA polymerase 3, which can be seen here, starts at the RNA primers and works in a 5' to 3' direction. In the leading strand's case, working continuously. In the lagging strand's case, working discontinuously, or working in fragments. Either way, the DNA polymerase 3 is the one that adds complementary nucleotides, or the DNA nucleotides, in sequential order. Okay, A pairs with T, T pairs with A, C pairs with G, G pairs with C, and again in that 5' to 3' direction. DNA polymerase 1 in both cases, in both strands, would replace the RNA primers with the DNA nucleotides. You can see DNA poly 1 in this case actually taking the red fragments out and would exchange the red fragments with blue DNA nucleotides. Okay, again, it's going to pull out the RNA primers and it's going to replace them with DNA nucleotides. As soon as the DNA polymerase 1 exchange the RNA primers with DNA nucleotides, then your DNA ligase is going to come in and is going to seal the nicks. And so all it has to do is basically form those phosphodiester bonds, linking the hydrogen, or linking the backbone, linking those covalent bonds, and we have formed a continuous strand. Okay, we have formed a continuous strand in discontinuous fashion. Okay, this happens all the way along the DNA molecule until the next origin is reached. At that point, the entire DNA replication process is complete, and we have two new semi-conservatively produced strands of DNA ready to basically join a new cell or make cool things happen. See ya.